Chapter 17, got procedures for identifying pathogens and diagnosing infections. This is like clinical microbiology, what you use to identify things for your patients. So we have methods of identifying unknown microbes. We can use one type of method of identification is phenotypic. Phenotypic is we have your microscopic morphology. We have a fresh or stained microorganism from a specimen. You determine its shape, its size, stain reaction to different dyes, and the cell structure. I'll determine what it is. Or you have macroscopic morphology, what you see without a microscope, like the appearance of the colony, the texture of the colony, the size it takes on, its shape, the pigmentation of it, and the requirements it needs for the colony to continue growing. Okay, different nutrients in the environment. You have physiological and biochemical characteristics. That's detection or the presence or absence of particular enzymes or metabolic pathways. If you block a certain pathway, does it continue to grow? Certain ones do, certain ones don't. So we're identifying certain microbes. Genotypic, okay, genetics, what makes it up? Okay, assess the genetic makeup of the microbe. Culture is not usually necessary. Usually do a DNA analysis. It's very precise. It matches your DNA for nucleotide to nucleotide. It's an automated method done by computers, and you usually get quick results. Or you can do immunological, like analysis of blood sugar we talked about, like A, B, and O blood types with antigens and antibody reactions to that. You have to collect the specimen first. You have to get it. So sampling body fluids for suspected infectious agents. Results depend on the specimen that's collected. How you have to handle it, how you transport it, how you store it can be different for different types of microbes you suspect are in the sample. Aseptic procedures should be used. Aseptic procedures means no contamination. You're not introducing other microbes during your collection procedure. Okay. Specimen collection. You can use swabs. You can use sputum, okay, from the nasal cavity, saliva from the mouth. You can use blood. You can use a catheter for urine specimen. What else we got here? Scrapings from the heel with a scalpel, spinal tap, feces, vaginal swab or stick, skin surfaces also with a swab, throat or your tonsil area, or the nasal pharynx. All the different areas you can collect specimens out in different ways, different techniques. Okay. Now, guidelines for specimen collections. Let's look at this. Okay. There's different, it shows you the type of specimens, and it shows like a type of specimen could be an abscess of skin or membrane or decubitus ulcer. You see there's a lot of bedridden patients. Okay. These are, you debride the surface area, and you have a sterile wipe, suction through a sterile needle, and a syringe. Aspiration of the tissue, preferred to swab, transfer to an anaerobic transfer system. In case you don't want to expose the sample to oxygen. Okay. Right. Anaerobic cultures. Anaerobic cultures are cultures that survive in the lack of oxygen. Collect from deeper tissues below the surface. Use sterile syringe. Samples must be exposed to air. If exposed to air, it will kill the anaerobic bacteria. Okay, so makes transfer immediately in an anaerobic system to the lab. Collecting with blood. Prepare your skin with iodine so you don't transfer bacteria from the surface of the skin into the bloodstream with your needle. Okay, so prepare skin with iodine. Use a vacutainer or Bactic blood collection tubes. Inoculate the blood culture bottle immediately with it. Bone marrow transfer. Bone marrow specimen. Prepare the site for a surgical incision. Extract the sample with a special needle that penetrates the bone into the bone marrow. Most samples taken from the sternum or the ilium, so either from your breastbone or from your hip. Cerebral spinal fluid is you remove cerebral spinal fluid by a lumbar puncture. Okay. Place the fluid in the sterile tubes. Don't refrigerate. Transport immediately for lab for processing. Collect microbes from feces. Small specimen in a sterile container. Cover and transport it. The genital or urinary tract, you sterile swab the cervical mucus, swab the urethral membranes, or insert into lumen and twist. Okay, plate directly on the selected culture media with high CO2 atmosphere, or collect with anaerobic transportation swab system. Okay, respiratory tract and lower, you have the lower tract or you have the upper tract. In the lower tract, down deeper in the lungs, okay, have patient cough to loosen the phlegm and expirate that into a sterile cup for transport. Now, the patient should brush their teeth and rinse with water prior to any sampling. Get all the bacteria. If you're going to spit it up, again, you want the bacteria, the microbe from deep in the lungs. You don't want the ones present in the oral cavity. Okay, try to do your best to eliminate bacterial cavity prior to patient spitting up into the cup for the microbe. The upper respiratory tract, you cleanse and debride the oral cavity membranes, swab the area for the lesion. Throat cultures are taken by swabbing the posterior pharynx and tonsils or the inflamed site. Two methods for collecting urine. One is by mid-catch stream. In other words, you start to urinate, stop, put the bottle there and continue the urination. That's what's called a midstream catch in the stroke container. Or you can use a catheter inserted into the urethra. Okay, drained into the bottle. So the routes taken in specimen analysis. You have direct tests. Once you get your specimen collected, okay, the direct test is with a microscope, immunological, like we test for blood with antigens to antibodies, or genetic, test for different DNA. Cultivation of our sample, isolation, identification, okay, when we cultivate our microbe. Two categories of result. You got presumptive data, which we guess what it is, and we modify that conclusion, and confirmatory once it's confirmed in the lab. Right. So the overview of laboratory techniques, we had direct testing. Remember, direct testing was microscopic and macroscopic. Microscopic was gram staining, gram positive, gram negative, acid fast stains, fluorescent antibody stains, which we'll show a couple examples of those just coming up. Gene probes or the fish testing. With the macroscopic, we have the direct antigen type. We did the antibody antigen reaction in the serum, which caused agglutination. So we have direct antigen or DNA typing or rapid pathogen identification. Okay. Cultivation or isolation was the other way of doing it with the the direct technique. Test on the isolates. We have biochemical testing to do, serotyping on a slide, DNA, RNA sequencing, PCR analysis, making the DNA into smaller sections and multiplying those. Phage typing, where that round DNA can be inserted into different types of bacteria. Animal inoculation, we knock an animal, see how it responds, and antimicrobe sensitivity, how they respond to different antibodies in the lab. Okay. Now the patient. Patient, a point of care test. You have clinical signs and symptoms of your patient. In vivo tests for reaction to microbe. You isolate the microbe and you test it in vivo and see how it's going to respond. Now you know how to treat your patients. Immunological and serological tests for the antibiotic titer are performed on blood and other fluids. Immediate direct examination, we can use a microscope. Okay, you can different stains, gram positive, gram negative stains. Acid fast stains, direct immunofluorescence antibodies, where we attach a fluorescent marker to the antibody. When that attaches to the antigen, wash out the rest of it, and the stuff that's still bound to it, well, it's going to glow when you see it under the microscope. Or direct antigen testing. Okay. So this is your exudate from your lesion, which contains your microbe. Okay, this is you attach a fluorescent molecule. These little nuclei are fluorescent. We attach that to our antibody, and then the antibody bonds to the antigen, and it keeps the fluorescent attachment with it. So we wash this off, and if there's no glowing from the fluorescent antigen attached to the antibody, then we know it didn't bind to it. So we know it's not that. We identify it's not that particular type of pathogen. Okay, so the syphilis spirochete binds to that with the fluorescent antibody, and it glows. So we know what type it is. And that's what it looks like under the microscope. That's your fluorescent. That's your syphilis spirochete. Okay, we have cultivations of a specimen. Isolation media, we have an enrichment of the media. We have a selective or differential media, depending on how what we want to use, see what type of a microbe it is. Different microbes are going to grow in different media. Biochemical testing, physiological reactions to nutrients. Some bacteria need certain specific nutrients or other substrates as indirect evidence of the absence or presence of enzymes. It breaks down certain proteins that you put in there. If different microbes produce different enzymes, we can help to identify different microbes that way. Unknown microbe and the growth in the microbe. Is the enzyme present in the microbe? You get a positive result. If the enzyme is absent in the microbe, you get a negative result. So if you put in a substrate that you know is reacting with a certain enzyme, you can tell that enzyme is being produced by that microbe. You get a positive result, you know it's being broken down. You get a negative result, you know it's not being broken down. And you're going to further eliminate and help identify which type of microbe it is you're dealing with. And the different ones you can look at, you get different wells, and you can identify different microbes by the substrate that they break down. It's usually a color reaction or precipitate or an agglutinate reaction, whatever we're looking for. Okay, phenotypic methods. We have miscellaneous tests, where morphology, which is just the shape of the microbe, and biochemical tests are not definitive. Okay, then you can do specific DNA testing, identify each one specifically, like phage testing. Phage testing, if you have phases of the circular DNA, we can put in there and see what it produces inside that microbe. Staphylococcus and Salmonella are two types we use for phage testing. Animal inoculation, Mycobacterium leprae and Treponema pallidum, we use for animal inoculation, and antimicrobial sensitivity, important to determine treatment of the drugs that we use. Okay, expose to different antibiotics in vivo and see how it reacts. Identify certain species of Streptococcus and Pseudomonas and Clostridium. All right, so cocci. Cocci are the round circular bacteria. You have gram-positive. Okay, gram-positive or the catalyst positive are irregular clusters and tetrads. Tetrads are four. Those are four units together. They're strictly aerobic, Micrococcus. Facilitative anaerobic are your Staphylococcus and Planococcus. And the catalyst negative are those are your chain arrangements. Those are Streptococcus and Enterococcus. In the gram-negative, you have aerobic and you have anaerobic. Okay. Aerobic is the oxidase plus and catalyst plus. Neisseria, Brenhamella, and Moraxella. Yeah, a type of bacteria or microbe associated with that. Anaerobic, oxidase plus and catalase plus are Veillonella. And the different bacilli. Gram-positive bacilli. You have spore formers like Bacillus clostridium. You have non-spore formers, which could be like acid fast for their staining, Mycobacterium nocardia, or non-acid fast for staining. You have regular ones like Lactobacillus listra, and pleomorphic, different types of it, is Corynebacterium, Propionibacterium. The gram-negative. Okay, aerobic, oxidase plus, and they're not curved. They're straight back microbes. Pseudomonas alcagines, and then you have facultative anaerobic oxidases negative, and they ferment glucose, break down glucose. They're modal. Okay. Escherichia, Enterobacter, Citrobacter, Proteus, Salmonella, and Erwinia. Okay, the non-modal ones, okay, non-modal ones that like flagellum or some kinds of self-propulsion, Shigella and Klebsiella. The aerobic oxidase plus, the curviform shape, okay, so these have a curve to them with their morphology, and they also ferment glucose. Vibrio, and ones that do not ferment glucose, do not utilize glucose as an energy source, is Campylobacter. Genotyping message, analysis by DNA. So you got DNA analysis with genetic probes. We use labeled probes with either single-stranded or double-stranded, which are single-stranded DNA RNA, and is complemented to a specific sequence of a particular microbe. Fluorescence in situ hybridization, which is your FISH technique, which is you're going to match your complementary DNA to this probe, and this probe is going to have a fluorescent marker attached to it. so we can see when it attaches after we water rinse everything off. And we can also use a pulse-generated electrophoresis. Remember electrophoresis? You put your proteins on it, and it separates the protein, separates the DNA into different bands. We use that in DNA analysis, forensics, and things like that. And it's just the larger molecules, larger DNA sections, move slower than the smaller ones. Smaller ones move faster. They're pulled towards the opposite pole faster. Okay, so your positive blood culture, you put on the Gram stain, and it results either Staphylococcus or Staphylococcus aureus. You're not sure? Then we put a fluorescent molecule attached to an RNA for Staphylococcus aureus. So we do a specific probe for a gene we know the sequence of in Staphylococcus aureus. So if it binds to it, we know it binds to that particular sequence, and it has this fluorescent molecule attached to it, so it glows. So you put it on there, and the parts that attach now are fluorescent after you rinse off everything else. And if we have under a microscope fluorescent activity, fluorescent, you can see fluorescence under the microscope, it is a test positive for Staphylococcus aureus. So we're identifying what type of microbe you have. The polymerase chain reaction, again, if we use with DNA, we talked about it before. It's most valuable for DNA and RNA analysis. It's highly sensitive and very specific. That's when we have like a crime scene with forensics. We have a little bit of DNA present. We can use these polymerase chain reactions to take the small section and duplicate it and repeat it and repeat it and repeat it and make larger, larger. So you have a larger sample size, so we can use that in our DNA analysis. Okay, so it's very highly specific, binds to a specific set of nucleotides. Okay, and it's very sensitive. It's used to detect, we use that in detection for HIV virus, for Lyme's disease, for human papillomavirus, tuberculosis, and hepatitis. Ribosomal RNA sequencing, studies of phylogeny and taxonomy, how they're related to one another with different microbes and how they are, what's what I'm looking for? Not environmental, but evolution related to each other, how they've evolved along the similar paths. So we studied the phylogeny and taxonomy of samples from complex microbes and environmentals that are using difficult to study. We can study their DNA and how it's related to different species. And we can match that way. This is electrophoresis plate. You can see the different bands being marked here. You can see the different band where they occur in the same ones, in the same regions. So this here, this here, this here. You can see the bands, and then pull and separate the different proteins and different DNA samples by the electrical field that go through a gel. Like I said, the larger ones move slower, the smaller ones move faster. Okay. Serology. Serology, we did that. We talked a little bit about that with A, B, and O blood types. This is in vitro diagnostic testing of serum. We get visualizations. We actually see the clumping of antibody antigen reactions, which includes our precipitate, which precipitate by definition is mixing two liquids and the solid forms. That's a precipitate in chemistry. We have to see color changes or release of radioactivity. We do a radioimmunoassay on it. You can detect or identify an unknown antibody using a known antigen, or you can identify a certain antigen using an unknown antigen using known antibody. So either way, it's a very high degree of specificity and sensitivity, since that antibody only binds to one particular antigen. So here's your antigens that are in the sample, and your antibody is only going to bind to one of the particular antigens here. This IgE antibody. Okay. So this red antibody binds with these antigens. The blue antibody here, IgE antibody, binds with the little triangles. It fits into there. They're stereospecific by shape. So it binds to different particular shapes onto the antibody. Okay. Now, if the antibody doesn't, like, here's your antigen, all these other antigens don't bind. They're not going to match the shape of the antibody present. So testing with blood, you can see the molecular reaction where the antibodies group together, because you're bonding them together that way. So in serological diagnosis of disease, a blood sample is tested for the presence of an antibody using an antigen of known specificity. You know the antigen is going to bind to a specific antibody. A positive reaction is a visible sign, like we talked about color change, clumping, agglutination, precipitation. Okay, and that shows there's been a reaction between the antibody and the antigen. So you can identify what antigen is present by knowing putting in a specific, but you know what antibody is present by putting in a specific antigen. Okay? And this is your testing. You take the different viruses, the different microbes you want to test for, and you mix them with a known antigen. And you can tell if they're present by the type of reaction that occurs. You can see the agglutination here and here, where it get clumps, whereas everything else is clear here with no color change and nothing else. So you test it on a sample, you take different wells on a card, antigens in a positive control, and the clumps indicate precipitated antigens, possible infection in the patient. So we know what antigen is there because we put in specific antibodies. So we can identify the antigen in your patient. Again, unknown micro is mixed with a serum containing antibodies of the known specificity. That's our serotyping. Okay. Macroscopic or microscopic clumping indicate there's a correct match. Okay, so you see the antibody here, and it's binding to an antigen on the surface of the microbe. And remember, you got two binding sites on your antibody. So it binds here, and this creates a clumping together. This is a molecular reaction as they group them together. And that's what forms your agglutination, which is makes you clumping as it's together. And that's a positive test for the presence of a specific antigen. Okay. On this side here, you see no clumping in one of those. On this side here, you see all this agglutination is clumping together. This side is a positive test for a specific antigen, so you know what kind of microbe it is. And this is a negative test for that same antigen. Yeah, and I didn't clump with it. Okay. So agglutination tests are the antibody cross-links a whole cell. So you have a bacterial cell with specific antigens on it. Okay, bacterial cell and specific antigens on it. The antibody binds to both those ends and it forms this clumping together. That's agglutination, where the whole cell is bound to it and the cells bind to the antibodies and they agglutinate. They form this mass in there. So it's antibody cross-linked whole cell antigens. So antigens that are on the bacterial cell themselves, forming complexes that settle out and form visible, insoluble clumps, which means they're not dissolvable. This is what we do with blood typing. We do our A and B blood types. Some bacterial and viruses are diagnosed this way also. And this is our, what we just talked about. This is the whole cell. Remember, epitopes are just the expression of the antigen on the surface of the cell. Okay. You get your specific antibody to it. Antibody has two binding sites, so it links these cells together into an agglutination, and it causes a mass appearance. Okay, precipitation is different than agglutination. So agglutination is your whole cell. Okay, so and it binds to that. Precipitation is remember, precipitation definition is two liquids that come together and form a solid. So you have an antibody antigen reaction, and it glues together. The big difference is this is not whole cell. This is not the cell. This is a whole cell agglutination. These are just antigens. Okay? They're soluble matter. They have to be dissolved in water. Whole cell is not dissolved in water. If you have a precipitate, you have to have two liquids mixing together. So it has to have a soluble protein or soluble antigen. So agglutination. For the question?
Yes, I have a question. For the whole cell, is that just blood?
No, it could be red blood cell in our serum test, or it could be the microbe, could be bacteria. It could be whatever we're testing for. It's a whole cell with the antigen expressed on the cell membrane.
So whatever we're testing, we have like a sample of that, and then we just use a cell of that to.
Right. Well, there's a whole bunch of cells in there in our sample. The microbe is present in our sample.
Oh, okay. And then we just add the antibody to it. Okay.
Got it.
Okay.
So in a agglutination test, you use a whole cell. In precipitation, use only the antigen. This is the epitope. It's not attached to the cell. It's soluble in the liquid, in the media, whereas whole cells are not soluble. Does that make sense?
Yes, thank you.
Okay. Perfect, no problem. Okay, so agglutination, we dilute it until everything's just soluble, but like 1 to 20, 1 to 40, 1 to 80. The more we dilute it, the less precipitate or less agglutinate that we have at the bottom, because there's less antigen to bind. Okay. So looking back at this picture here, okay, we got the tube agglutination test. A sample of the patient's serum is serially diluted. So 1 to 20, 1 to 40, 1 to 80, 1 to 60, and so on. Okay, with saline, sterile saline. Dilution is made in a way that halves the number of antibodies in each subsequent tube. So an equal amount of the antigen, which is your blue stuff that's added, cells that are added to each tube. The control tube has antigen, but no serum. Okay. After inoculation and centrifuge, each tube is down for agglutination and compared with the control, which will contain the unagglutinated cells and no clumps. So the titer is equivalent to the denominator of the dilution and the last tube in the series that shows the agglutination. So it tells you how much of the antigen is present for the infection to occur as each serodilution occurs. So we talked about the agglutination test. That was that. Precipitation test is a soluble antigen. It's not the whole cell. Remember, it has to be soluble because it's two liquids that are being formed together. Once you add the antibody, and it makes it, once you add the antibody, then it binds to it and causes this solid to appear. That's the precipitation test. So you're testing with two different things. And you have this is for a VDR, this is a Venereal Disease Research Labs, VDRL. Most precipitants are done in the agar cells. Okay, so this is your antibody antigen. This is your precipitation. Okay, you can see how you added into each cell here. This is on the slide. Okay. You have different areas where you test each one. You have a control, you have the antigen, then add the serum to it, and then this is what your actual test results look like when you examine it. You can see which ones have reacted, which ones have not formed to precipitate. So agglutination reaction, the antigens are what? Precipitation reaction, the antigen is what? So agglutination,
Yeah.
the antigen is the whole cell, right?
Yes.
All right, and precipitation, the antigen has to be solid. You said B, right?
Yes.
Perfect. You got it. So agglutination is the whole cell that reacts to the antigen on the cell membrane. In the precipitation, it's just a soluble molecule of the antigen. Western blot technique detects proteins. This is our electrophoresis. Remember, electrophoresis is our gel plates with the charges, positive charge applied to both ends, and the movement of these proteins are due to their size being moving quicker across the gel medium. So we have electrophoretic separation of proteins, followed by an immunoassay to detect these proteins. We bind them to immune compound to identify them, and those will speak to, they're highly specific and sensitive. So the electrophoresis separates them, then we take that band and we run it through RNA markers that bind, or DNA markers that bind with a complementary DNA, and it's very specific and binds them to a specific genetic pattern of the nucleotides. And this is a second test used to verify HIV status for AIDS. And it's a site-specific antibody binding will appear as a pattern of bands. That's your electrophoresis. So here's your electrophoresis. Here's the different bands we use. Then we use each of these bands that are separated and run through an immunoassay to identify which one each band is. So successive tests on an HIV positive patient over 30 days reveals an increase in band intensity over time due to the increased formation of the anti-HIV antibodies. You run through the plate, you have more antibodies with time as it develops. Test based on lysine mediated hemolysis. Lysine, antibody that requires the actions of a complement to complete the lysis of its antigenic target cell. So the test used on some viral, rickets, fungal infections. It often replaces by more sensitive and accurate what's called the ELISA test and genetic tests. Okay. So test antigens react with test antibodies, just like everything else. And the contents of tube 1 are mixed with sheep red blood cells. Complement used up in the first stage, there's no hemolysis. It's an unfixed complement. Then you get hemolysis. Okay, so you have stage 1 and stage 2. Stage 1 is your positive antibody antigen reaction. Okay. Then we get the complement, this fixation of this complement. You need this for lysis to occur. If you don't have that complement, okay, no lysis occurs of the cell. Once a complement attaches, it binds to this, and then you can get once it gets the complement attached, the complement can attack the red blood cell and cause lysis or destruction of the red blood cell. Okay. So just some of them need a complement in addition to the antigen antibody reaction. Okay. And that's your ELISA test. Okay. Serological test, Treponema pallidum immobilization is for syphilis. Okay. This is your toxin neutralization. You get serotyping for identifying, classifying, and subgrouping it into categories. Are your serotypes using the anti-seria for cell antigens such as the capsule, the flagellum, or the cell wall. Okay. Typing of salmonella. Okay. Our first piece is saying identification of stereotypes of streptococci. We use for that. Quolling test identifies serotypes and pneumococcus. In vivo testing, the antigens are introduced directly into the body to determine the presence or absence of antibodies to see if you get the reaction. That's your tuberculous skin test. Allergy testing, we introduce the allergens directly into the body. And we show the different reactions that can occur when the antibody reacts to the antigen that we put in. The fluorescent antibodies we talked about, monoclonal antibodies live by a fluorescent dye. Okay, an unknown test specimen or antigen. Okay. Fixed to a slide, exposed to a fluorescent antibody. Okay. Identify antigen on the surface of the cell. Okay. And if it binds to the cell, then the fluorescence stays after everything else is rinsed off, and it glows with that. That's used to diagnose syphilis. You should have syphilis, gonorrhea, chlamydosa, whooping cough, Legionnaire's disease, the plague, trichomonas, meningitis, listeria. Okay. And all that is your this is your fluorescent glowing with the antibodies that adhere to the antigens. Fluorescent antibodies are made to react with the FC region of another antibody, which is your constant region, which is not your binding site for the antigen. So the fluorescent part does not, think of your antibody. Okay. Remember, this is where you have your binding site. If the fluorescent marker bound there, you couldn't bind the antigen. So the fluorescent marker has to bind to the FC portion, to the constant portion. This is where we bind our fluorescent. So the binding site is free to attach to a antigen. We use these immunofluorescents to diagnose syphilis and various viral infections. Okay. So if the antibody is found in the antibody in the serum, the antibody binds to the microbe, or if the antibody is not in the serum, it doesn't bind. So this would have fluorescence. This would not have fluorescence, because the antibody doesn't find the antigen that it binds to. We can identify the different type. And this is syphilis, as you're looking for the different fluorescent glow of the antibodies attached to the antigen on the syphilis microbe. Immunoassays, we talked a little bit about them, extremely sensitive because we use DNA sequencing to attach to it. Extremely sensitive to take antigens and antibodies. Antigen antibodies labeled with radioactive isotopes. Okay. Then once we have the reaction occur, we run it through a Geiger counter, and it counts radioactivity and it tells us how much of it is bound to that section. So enzyme-like immunosuppressant assay is ELISA, we talked about. Enzyme antibody complex produces a colored product when an enzyme reaction occurs. We have an indirect and we have a capture of those antibodies, and we can count them. So indirect, compare positive versus negative reaction. This is what we use to test for HIV. We have the two different wells. Okay. You see in one of them, the antigen absorbs the antibody, the other one doesn't. When you rinse it off, this antigen stays. Okay. So we use our markers here that this binds to a certain enzyme, and the enzyme is positive if it binds to that area, and we use that to identify certain proteins that react with that enzyme. Whereas the antibodies don't bind, it's a negative result. Okay, capture or antibody sandwich, okay, is what the antibodies do. The antibody traps your whole bacteria cell with the antigen, and then the enzyme attaches it to the top of it, and it basically squeezes the microbe between them and results in a positive test. Okay, once the antibody binds. Once you rinse it off, if the enzyme doesn't bind to the microbe because the antibody wasn't attached to it. It doesn't trap the microbe so the enzyme can attach to it. These are the different plates look like. These are the different positive tests, and the clear ones are your negative tests. So in virus diagnosis, the virus presents special difficulties because they're not cells. Remember, virus is just a protein coat with a DNA or RNA strand inside of it. Viruses are labor intensive to cultivate in the laboratory. Techniques available to diagnose viral infections include observation of symptoms. Sometimes you just use a symptom of the patient, say it's a viral infection and what type it is. Direct microscope examination of the virus. We can cultivate it. Serologic, we can test for it, analyze the blood, or we can test for the DNA strand inside the protein coat of the virus. And these are different viruses in which we do diagnosing of them. This was the Elisa method we talked about. This is a Western blot for HIV. This is we can do cell cultures, electron microscope by direct visualization of it, fluorescence for syphilis scanning, clinical evaluations. Herpes virus is identified clinically. Okay, and we do the electrophoresis separation also. All right, so the Western blot test. Remember that was when we used electrophoresis. We have the different panels. We took that one band that was isolated and we used that to match with its DNA and specifically identify the virus or the microbe by using a DNA probe. Right? So it can be used to identify unknown antibodies, unknown antigens, specific DNA, or both A and B. Now remember the test, the whole test is based on this electrophoresis, which is the separation of proteins by size and charge. So DNA is not a protein. It's not going to respond to the electrophoresis that we're doing for the Western blot test. Antibody is a protein and antigens are proteins. So it would have to be A and B. It would have to respond to the proteins being moved through the electrophoresis. And then we separate with the DNA. So the Western blot can be used to identify unknown antibodies and unknown antigens. Does that make sense?
Yes.